Our next speaker is Dr. Eric Freed. Dr. Freed received his PhD in 1990 from UW-Madison, working with another well-known McArdle faculty member, Rex Risser. He then performed a postdoc with Howard Temin, and shortly thereafter he joined the National Institute for Allergy and Infectious Disease, NIAID, as a staff fellow. A few years later, he was appointed as a tenure track investigator at NIAID, and he received tenure in 2002. 1 year later, he joined, as we heard from Dr. Coffin, the HIV Drug Resistance Program, now named HIV Dynamics and Replication Program, within the National Cancer Institute. 2015, he was appointed as the director of the HIV DRP. He's also co-director of the virology program at University of Maryland College Park, and he serves as editor-in-chief of the journal Viruses. Today, Dr. Freed will talk to us about his journey from murine leukemia virus to HIV, 40 years in retrovirology.
Thanks very much. Can everybody hear me? I'd like to thank Paul and Paul and the other members of the organizing committee for putting together this really fantastic event and for inviting me to participate. I'm really honored to be here. I realize I'm the only thing standing between you and lunch. So in the next hour and a half, I'm going to try to tell you a little bit about how I got to Madison, what I did after I left, and I'm going to tell you about an ongoing project in the lab that you might find both surprising and interesting. So I did my undergraduate work at Penn State, and during my coursework, I became really fascinated by viruses, in particular retroviruses. So this was during the time when Harold Varmus and Michael Bishop were uncovering the relationship between cellular proto-oncogenes and acutely transforming oncogenes carried by retroviruses. This work, of course, was made possible by the really groundbreaking work of Howard Temin decades earlier. So I was fortunate as an undergraduate to work in the lab of David Shalloway, studying the cooperation between oncogenes. In 1985, I came to Madison with the goal of working on retroviruses. Here's the old McArdle building, and I think you can date this. Well, this is going to be a little challenging to use this pointer. Stop working again. Okay, that makes my life easy. There we go. Okay, there we go. If you look at the cars in the parking lot, this is probably a 1970s vintage, and I love to cross-country ski, so Madison was a good place for me. So I landed in the lab of Rex Risser, who was a young professor in McArdle, studying transformation by murine leukemia viruses. My committee was made up of Howard Temin, Bill Sugden, Roland Ruckert, and Colleen Hayes. And so the first project in my lab involved studying the role of envelope glycoprotein processing in murine leukemia virus fusion and infection. This was a bit of a divergence from what was ongoing in the lab, but I found viral envelope glycoproteins to be particularly fascinating. And this was during the early years of the AIDS pandemic. And so I really felt a calling to switch my research from murine leukemia virus to HIV. I continued the study of the HIV envelope glycoprotein, mapping out domains involved in glycoprotein processing, fusion, virus entry. And my plan had been to finish my PhD in 1990 and then do a one-year postdoc with Rex, while my then girlfriend and now wife, Janelle Kortner, finished her PhD with Peggy Farnham in McArdle. But very tragically, Rex Risser passed away in the fall of 1990 at the age of 42. So Howard Temin took me under his wing, and I really had to say that the faculty of McArdle did a really phenomenal job of taking care of Rex's students ensuring that we would continue along our career paths as uninterrupted as was possible given the circumstances. In Howard's lab at the time were a few people who were sitting around this table. Here I am with Wei Su and Wen'ei at a Cold Spring Harbor retrovirus conference. I think it was actually RNA tumor viruses still at that time. And here I am again at a wine and cheese party, which is when they took most of their photographs, with Lou and with Kathy. And you'll be hearing from them later in the day. Also, in 1990, we already heard about this event. Cold Spring Harbor held the 20th anniversary of the discovery of RT. Here on the left, my pointer's not working, but here on the left is Howard enjoying a piece of RT birthday cake. And on the right, Howard with John Coffin and the late, great David Baltimore. And again, we're all very sad to hear the news last weekend that David passed away. So in 1992, I started a staff fellow position, basically a glorified postdoc position at NIID in Bethesda. I was on tenure track from 97 to 2002, received tenure in 2002. And during this time, I continued to do some work on envelope glycoproteins and virus entry, and also nuclear import, a little bit about which you already heard. But my main research focus shifted to studying the late stages of the HIV-1 replication cycle: virus assembly, envelope glycoprotein incorporation, virus budding, and virus maturation. And then in 2003, as was mentioned in the introduction, I moved my lab to the HIV, then drug resistance program, now dynamics and replication program. And you'll see in the front row, again, as was already mentioned, several of today's speakers, Wen'ei and Wei Su, and John Coffin, the founding director of the DRP. The baton from Steve was handed off to, John was handed off to Steve Hughes, who's at the far right, and then to me in 2015. So I'm going to now shift to tell you a little bit about some of the work ongoing in my lab. But first, I'm going to give an introduction to where we stand, or an overview of where we stand with the AIDS pandemic. So AIDS has killed around 44 million people worldwide since the beginning of the pandemic, and a similar number of people are currently infected with HIV-1, currently living with HIV-1. Combination antiretroviral therapy, or ART, which primarily targets the three viral enzymes, reverse transcriptase, protease, and integrase, has greatly increased the lifespan of people living with HIV. However, while the prognosis for people with HIV is much better now than it was 20 years ago, the HIV problem is far from wrapped up. Suppression by antiretroviral therapy blocks replication but does not cure the infection, so lifelong therapy is required to treat people living with HIV. As a result of this increased lifespan, the number of people living with HIV globally actually continues to increase. And long-term antiretroviral therapy and low-level viremia are associated with a variety of serious adverse effects, including chronic inflammation, kidney, liver, and neurocognitive disorders, and cancer. And poor adherence and inadequate viral load monitoring increase the risk of drug resistance, which is rising in many parts of the world, particularly in resource-limited settings. So this slide shows the HIV replication cycle and the steps that are currently blocked by approved antiretroviral drugs. Again, primarily the three viral enzymes, RT, integrase, and protease. We also have entry inhibitors and a newly approved capsid inhibitor known as lenacapavir that many of you have already heard about. And so by and large, resistance to these drugs emerges when the virus acquires mutations in the proteins that are targeted by these drugs. In other words, HIV becomes resistant to RT inhibitors by acquiring mutations in RT, and so on. In the case of integrase inhibitors, though, the situation is a little bit different, as I'll describe for you over the course of the remainder of my talk. So what are integrase inhibitors? So these are, the approved drugs are integrase strand transfer inhibitors, or INSTIs. They bind what's known as the intosome, which is a complex between a multimeric form of integrase and the viral DNA ends, to block the strand transfer step in integration. INSTIs are highly potent and difficult for HIV-1 to evade. Although integrase mutations do arise in some INSTI-treated individuals, these mutations generally confer fairly low-level resistance, and quite interestingly, virologic failure, that is the emergence of resistance in vivo, can occur in the absence of mutations in integrase. So this latter finding indicates that mutations outside integrase likely contribute to virologic failure to INSTI-containing regimens. And what I'll tell you about in the remainder of the talk is that we found that mutations in envelope and the nucleocapsid drive escape from INSTIs in cell culture. And we're finding now very recently that NC mutations, including the same mutations that we obtained in cell culture, arise in people with HIV failing INSTI-containing regimens. So Yuta Hikichi in the lab asked, how does HIV-1 develop high-level resistance to INSTIs? So he performed long-term passaging of HIV-1 in a dose escalation scenario in the presence of the INSTI dolutegravir. He used multiple HIV-1 isolates, including transmitted founder viruses that are clinically relevant, as well as not only T cell lines, but also primary T cells. And this is an example of one of these experiments. So on the x-axis is the passage number in weeks. So this is a nearly year-long experiment. And on the y-axis is the dolutegravir concentration in nanomolar on a log scale. The IC50 for dolutegravir in the system is about 0.5 nanomolar. So over the course of this nearly year-long selection, Yuta was able to increase the concentration of dolutegravir up to 4 micromolar, or more than 3 logs over the IC50 of the inhibitor. And then he sequenced the virus as he went along, as indicated by the numbers and colors, which you don't need to pay too much attention to. But basically, the take-home here is that HIV-1 acquired, in a sequential fashion, mutations in envelope, nucleocapsid, and integrase. And in most cases, the integrase mutations that he selected for conferred only low-level or no resistance to INSTIs, indicating that it's really the mutations in envelope and/or nucleocapsid that is driving the ability of the virus to replicate at these very high concentrations of the drug. So because of, the selected integrase mutations typically confer low-level resistance. I'm just going to now summarize some work that's been published because of shortness of time. So Yuta found that the accumulation of multiple envelope mutations leads to high-level INSTI resistance. The envelope mutations work by enhancing the ability of the virus to spread efficiently, effectively increasing the multiplicity of infection, or MOI. And I'm reminded, John mentioned about Cold Spring Harbor, people asking Howard's question. One of the questions that he always asked at Cold Spring Harbor was, what's the MOI? So I think these results really indicate that MOI can be extremely important, and often virologists ignore the multiplicity of infection. So the envelope mutations confer resistance to other classes of antiretrovirals, but interestingly, they do so at a markedly lower level than they do to INSTIs. This raised the question of whether INSTIs are inherently more susceptible to increased MOI than are other classes of antiretrovirals. And the answer to this question is yes. I won't have time to show you the data, but they're in the paper listed at the bottom right. So the reduced activity of INSTIs at high MOI drives the virus to acquire mutations in envelope that increase the MOI. So why are INSTIs inherently more sensitive to high MOI than other classes of inhibitors? We think this is a result of the mechanism of action of these inhibitors. So for example, in the case of RT inhibitors, there are thousands of catalytic steps at which RT inhibitors can work during the infection process and block productive infection. In contrast, as I mentioned earlier, INSTIs bind the intosome, this complex between integrase protein and viral DNAs, to block a single catalytic step in the infection process, that is the strand transfer reaction. So INSTIs act during a narrow time window between completion of reverse transcription and viral DNA integration. This will become important in the last few slides. So as I mentioned earlier, HIV-1 acquired mutations not only in envelope, I've told you just a little bit about that, but also in nucleocapsid. Those are circled here. The NC mutations were not selected during propagation in other classes of antiretrovirals. So this acquisition of NC mutations is truly specific for INSTIs. So as some of you know, the NC domain of GAG, which is a zinc finger domain containing protein, promotes RNA packaging into virions during virus assembly. And the mature NC protein is a nucleic acid chaperone that stimulates post-entry events, including reverse transcription and possibly integration. And so, as mentioned, we selected for a number of mutations in NC that confer modest levels of resistance to INSTIs. This is shown here on the graph on the right. This is the dolutegravir IC50, so the higher the IC50, the less susceptible the mutant is to the inhibitor, and the mutants are shown along the x-axis. So one slide to tell you what the NC mutations do not do. They do not interfere with GAG processing, virus assembly or release, or RNA packaging. They don't impair virus replication or particle infectivity. They don't substantially alter the formation of viral DNA ends. If they did, that would affect the ability of the drug to bind and block integration. They don't alter the kinetics of uncoding. This is work done in collaboration with Ryan Burdick in Wenei's lab. And they don't substantially alter integration site selectivity. But what they do is they accelerate the kinetics of viral DNA integration. So we can do time of addition experiments with RT inhibitors and integrase inhibitors, and we can determine essentially the time at which the virus becomes insensitive to these inhibitors. And when we do these experiments with wild-type HIV-1, we find that there's about a 3-hour lag between the completion of reverse transcription and viral DNA integration. And when we do the same analysis in parallel with these NC mutations that confer resistance to dolutegravir, we see a statistically significant shortening in the time window between completion of reverse transcription and integration. So the NC mutations selected in the presence of dolutegravir shorten the time between the completion of reverse transcription and viral DNA integration, narrowing the time window, the window of opportunity during which INSTIs can act to block viral DNA integration. We also observed that the nucleocapsid and integrase mutations cooperate to increase resistance to INSTIs. So shown here in the graph, single amino acid mutations that we selected for in NC and integrase confer around threefold resistance to dolutegravir. If we combine the NC and integrase mutations, we see about an 8 to 9-fold level of resistance to this inhibitor. So of course, a key question is, do mutations in envelope and/or nucleocapsid contribute to INSTI resistance in people with HIV, or is this just a cell culture phenomenon? So we're currently working with several groups to analyze samples from individuals failing INSTI-containing regimens, in many cases without mutations in integrase. And this is a list. This list continues to grow of clinical collaborators with whom we're working on this project. And just a little bit of recent progress without showing you the actual data. Participants in this particular ACTG clinical trial failing a DTG-containing regimen with verified drug levels acquired mutations in NC, some of which were the same NC mutations that we picked up in our cell culture selections. And work that we're doing with a group in South Africa has also observed NC mutations in people with HIV failing a DTG-containing regimen. Again, some of the same mutations that we selected in cell culture. So to summarize, HIV-1 frequently acquires multiple mutations in the envelope and nucleocapsid coding regions during long-term propagation in the presence of insties. These selected envelope mutations confer resistance to insties by increasing infection capacity. They accelerate the acquisition of additional drug resistance mutations outside envelope. The NC mutations reduce the time between completion of reverse transcription and viral DNA integration, potentially limiting the window of opportunity for insties to bind endosomes and block integration and infection. NC and integrase mutations cooperate to increase NC resistance. The NC mutations do not confer resistance to other classes of antiretrovirals, so this is truly specific for this class of drug. Together, these findings demonstrate that mutations in multiple regions of the HIV-1 genome, specifically envelope, nucleocapsid, and integrase, collectively contribute to NC resistance in cell culture, and NC mutations, including some of those that we identified in cell culture selection experiments, arise in people with HIV failing NC-containing regimens. So I think a take-home from this work is that in cases of virologic failure to NC-containing regimens, particularly in the absence of mutations in integrase, we need to look outside integrase for the cause of virologic failure. So with that, I'll acknowledge people in my lab who have been involved in this work, particularly Yuta Akichi, who's really been the driving force behind this work, other groups in the DRP, including Renee's group, and our clinical collaborators. And finally, I'd like to acknowledge my former Madison mentors, Rex Risser and Howard Temin. It's really a shame that they can't be with us here today to enjoy this wonderful event. I'd also like to acknowledge my PhD thesis committee members, Howard, Bill, Roland, and Colleen. And if there's time, I'm happy to take any questions.
Do you have any data on the susceptibility of the nuclear capsid mutations to the so-called broadly neutralizing antibodies and to other specific antibodies?
Yeah, we've not looked at that specifically, but we have looked at those mutants in the context of virus entry inhibitors. And we see that they have no effect on virus entry, as you would expect. So this is really clearly the role that the NC mutations play is clearly a post-entry phenomenon.
Hi, Eric.
Oh, yes, Michael.
Yeah, that was great.
Thanks.
You might have mentioned this, but is there a cost to the NC mutations in the absence of dolutegravir?
So these mutations that we selected in cell culture are highly fit. They don't affect virus replication kinetics. One sort of caveat to that is some of the NC mutations that are now being picked up in clinical samples do seem to reduce viral fitness. And what we're seeing in the sequences from the South African cohort is we find very complex mutants that have many changes in NC. So it may be, as happens with other drug targets like protease, there might be some mutations that confer resistance and others that fix the fitness cost imposed by those mutations.
Hey, Eric, fascinating. So mechanistically, what do you think those changes are doing to facilitate that speed in transition, since it doesn't seem
right. So
obvious?
as I mentioned, NC is a nucleic acid chaperone. It's involved in RNA and DNA condensation. So we think that our working model is that NC binding to the viral DNA ends affects the ability of integrase to recognize that substrate for integration, and these mutations somehow change that. But we've got experiments underway to directly test that hypothesis. That's our working model.
Do you see any difference in where the virus integrates?
No, we don't. So we did integration site selectivity, and there's no change.
Questions? Any other questions? If not, thank you.
Thanks.